Hello, my name is Katie Snell and I'm delighted to be here today to talk to you about gene therapy and the patient journey through hematopoietic stem cell gene therapy. So I'm a registered paediatric nurse. I've got 13 years experience working within the field of bone marrow transplants and hematopoietic stem cell gene therapy. 6 of those years were focused specifically on hematopoietic stem cell gene therapy research, and I currently work at Orchard Therapeutics, providing education and support to hospitals as they treat children with hematopoietic stem cell gene therapy. So an important part of the process is understanding how genes work and how we can use them to treat genetic conditions. So genes are small sections of DNA. Each person has 2 copies of each gene, one inherited from each parent. Genes act as instructions to make building blocks of the cell, enabling them to function normally. So a bit like a recipe book. A genetic mutation is a change to the structure of a gene or a group of genes. Many mutations cause no harm, but others can cause genetic disorders. And there are different types of gene therapy, but the aim of gene therapy is to use genes to treat diseases using different approaches. Gene therapy has been approved as a treatment for several rare inherited diseases, and there's much more ongoing research to find new ways to treat even more diseases. There are 3 main types of gene therapy. So there's gene editing, which is inserting, deleting, or replacing DNA at a specific site. So a bit like cutting and pasting. There's gene expression alteration, which is usually inactivating or knocking out a mutated gene that's not functioning properly. So sometimes genes can overexpress or work too much. There's gene addition or gene insertion, which is introducing a functioning copy of the gene or a working copy of the gene into the body to help treat disease. And that's what we're going to focus on today. So types of gene addition or gene insertion. There are different methods to add a gene, but they all use viral vectors. A viral vector is a virus that has been engineered to be non-infectious and non-disease causing. It's used instead to transport the functioning gene or the working gene into the cell, so a bit like a courier. The most commonly used viral vectors are adeno-associated vectors or AAV, and these are usually used in vivo gene therapy, where the therapy is administered directly to the patient and the targeted cells remain in the body of the patient. And then there is lentiviral vectors. These are usually used with ex vivo gene therapy, where the targeted cells are removed from the patient. The gene therapy is administered to the cells in a lab before they are returned back to the patient. So hematopoietic stem cell gene therapy is a type of ex vivo gene therapy. Stem cells are produced by the body and are used to make new cells when needed. Hematopoietic stem cells are our blood stem cells, and they are found in our bone marrow. These cells are capable of producing copies of themselves, which is self-renewal, and they produce blood cells of all types. So white blood cells, which help our bodies fight infection, red blood cells that carry oxygen around the body and remove the carbon dioxide, and platelets, which help our blood clot. They can also travel throughout the whole body, including the brain. And so if we look along the gene therapy or the hematopoietic stem cell gene therapy patient timeline or patient journey, there are specific milestones along the way, and we're going to talk through these specifically in the next few slides. So information given, that initial information time or meeting the team or the specialist gene therapy team, this is a really important part of the process. Any type of treatment or new treatment is really scary, and especially complex treatments like gene therapy or bone marrow transplants. There's a lot to understand. So spending the time with the specialist team, understanding the journey, there are lots of things to consider. There are other jobs, there are other children to think of. So making sure that if you're thinking about any treatment, that patient journey has been explained nice and clearly, and you understand that the opportunity to ask all your questions. So screening and eligibility for gene therapy. So any treatment or any medicine requires a period of eligibility and suitability assessment. Each patient will undergo a period of assessment to establish, is this treatment appropriate for them? Things that the specialist team may look at is disease state or disease progression, and general medical state, so things like heart and lungs will also be looked at as well. And then if we move on to the next stage, which is cellular source collection. So cellular source collection means to remove some of those hematopoietic stem cells or blood stem cells that are found in the body. Now these can be removed directly from the bone marrow, or more commonly now from a process called apheresis or leukapheresis. In bone marrow transplants, the blood stem cells are collected from another person, so the donor. And this donor may be a family member or someone unrelated that matches the patient. In hematopoietic stem cell gene therapy, the blood stem cells, as we said, are taken directly from the patient. This means that searching for a match isn't always needed, although sometimes it can be used to help support in decision making and discussions. And also, rejecting the donor stem cells is not a risk because you're your own donor in hematopoietic stem cell gene therapy. So collecting the stem cells using apheresis will usually involve a series of injections over a few days. These injections help move those blood stem cells or those hematopoietic stem cells out of the bone marrow and into the blood. During this time, the patient will be admitted to hospital for approximately 1 week. And once those blood stem cells or those hematopoietic stem cells have been collected, the patient is usually discharged home while those blood stem cells are modified in the lab. And then we can move on to the next stage, which is gene therapy manufacture. So those collected blood stem cells are transported to a specialist lab. And in the specialist lab, they select and purify those very early blood stem cells. They then use the viral vector to deliver the working copy of the gene into those very early blood stem cells. The patient's own stem cells then have a working copy or a functioning copy of that gene. And we can see from the pictures below on the screen that first picture, that first blood stem cell contains the white faulty gene. The second picture, we can see that viral vector that is delivering that blue working copy of the gene into the blood stem cell. And that third picture is the blood stem cell that's containing that white faulty gene, but also now that new working copy of that gene. So after manufacture of the gene therapy cells, these may be frozen to very cold temperatures, which is known as cryopreservation. Tiny samples of the gene therapy cells are tested to ensure they meet the quality and safety criteria to be able to give back to the patient, and also to check to see if they contain enough of the working copy of the gene. And then we can move on to the next stage of the patient journey, which is the gene therapy infusion. So the preparation itself and then the infusion. So a patient will be admitted to hospital for approximately 6 weeks. During that time, most of it will be spent in what we call protective isolation. Whilst in hospital, the patient will have very regular observations, and that's things like checking temperature, checking blood pressure, and very regular blood tests as well, looking at things like liver and kidneys and red blood cells and white blood cells. They will also receive a lot of supporting medicines as well. So then conditioning medicines, known as chemotherapy, are given to the patient to make space in the bone marrow and prepare the body for those gene therapy cells. This step of preparation with chemotherapy is also used for patients who undergo bone marrow transplants. And I like to think of it a bit like, pruning your garden. You need to get rid of some of the old plants or the old weeds, so that the new seeds that you plant are able to grow and flourish. And then it's the infusion itself. So gene therapy, hematopoietic stem cell gene therapy is usually a very, a small bag. Sometimes it's one bag, sometimes it's multiple bags, but it's very small, and there's a lot of emotions around this day, because as you've seen from the patient journey so far, so much work goes into getting a patient to this time point. After the gene therapy cells have been administered to the patient, we then follow a patient follow-up. So after chemotherapy, as we said, patients will be on protective medicines as their immune system grows. Once engrafted, those hematopoietic stem cells can continue to self-renew and give rise to blood cells of all types that travel around the body. By using the hematopoietic stem cells that have the ability to self-renew, the gene therapy approach has the potential to permanently treat genetic diseases. And after approximately 6 weeks in hospital, the patient is usually discharged. However, specialist teams will continue to follow up the patient for a long time, and this may be at specialist centres or a local hospital. They will look at things like disease state or progression, post-chemotherapy recovery, and engraftment. And specifically for gene therapy patients, they are followed up for at least 15 years, because gene therapy is a new type of treatment. And then I just want to finish on a slide about resources and support. Going through any type of treatment such as bone marrow transplants or hematopoietic stem cell gene therapy is a really hard thing to do, not just for the patient, but for the whole family. So utilising support systems where you can is so important. Friends and family, do invite them around to clean your house if you need, or bring family members to appointments with the doctors, especially if they're discussing complex topics. Or if they can't come to the appointments themselves, you can have them on a FaceTime or a video, WhatsApp video call. Utilize them as a support system. There is also various support within the hospital, so speak to your specialist team and see what support is available within your local hospital and your specialist hospital. And then there's also the patient advocacy groups. They are a great resource for education and support, and finding that community to help you through the treatment process. And so that's the end of my slides, but I would love to take any questions that anyone might have.